Hello friends, welcome to another video of SWA Environment Consultants and Engineers. This is video number 4 of Water and Wastewater Quality Testing Lecture Series Part 1. In this video, we are going to learn about the analysis of orthophosphate, total phosphate and total phosphorus in wastewater as per AFA 23rd edition method 4500P, which is of stannous chloride method. So let's start the video. So in this video, we are going to learn about, firstly we'll know the introduction or definition of phosphate and phosphorus. We'll gain some general information about phosphates and phosphorus. Then we will learn about the principle of measurement. Then we'll see which type of equipments will be required to perform the procedure. Then we'll see the procedure of analysis followed by the calculation and measurement uncertainty. So let's move to the introduction part. So let's start with understanding the definition and some general information about phosphorus. So basically the phosphorus found in water, soil, sediments, generally it does not found in gaseous form. So the phosphorus is highly reactive. That's why it always found in the form of phosphates. There are many type of phosphate available in the nature like ortho-phosphate, metaphosphate, pyrophosphate, polyphosphate, where the ortho-phosphate is the most common form of the phosphate available in the nature. The main difference between phosphate and ortho-phosphate, other phosphates and ortho-phosphate, is that phosphate is any compound composed of phosphate units. They are sometimes ester or salts of phosphoric acid, whereas the ortho-phosphate is composed of only one phosphate unit. The permissible limit of phosphorus in inland surface water is 5 mg per liter as per CPCB standards. In municipal sewage, the phosphorus level is generally between 5 to 20 mg per liter, in which 1 to 5 mg per liter is organic and rest is inorganic. Excess amount of phosphorus can cause eutrophication in water bodies. So what the eutrophication is, basically the water body witnesses the sudden bloom of algae on the surface. That's why the sunlight and oxygen cannot pass through. So the lack of oxygen and sunlight occurs in the water body, which eventually harms the aquatic life. High exposure of phosphorus in drinking water can cause the kidney problems, heart disease, and also it disturbs the metabolic system also. So this was the basic information about phosphate. Let's understand the principle of measurement for this procedure. So basically this procedure is a spectrophotometric procedure where the measurement of orthophosphate is carried out by spectrophotometric method at 619 nanometer wavelength. The color development is carried out with the help of ammonium molybdate reagent and stannous chloride. So basically what happens is, if the sample is containing orthophosphate and you add the ammonium molybdate reagent into it, the molybdophosphoric acid will get formed. In that molybdophosphoric acid, if we add the stannous chloride, the molybdenum blue will appear, which indicates the color development and presence of orthophosphate into the sample. In case of measuring the total phosphorus or total phosphate, the same methodology is used, but the sample is digested by using the concentrated H2SO4 and H1O3, which converts all different type of phosphate into orthophosphate. As we have understood in first slide, there are many types of phosphate available in the nature. So all other type of phosphate will get converted with the help of this digestion, and they will convert it into orthophosphate, and with the same method by adding ammonium molybdate reagent and stannous chloride, we can measure the total phosphate and total phosphorus too. That we will understand further in step-by-step procedure again. So now let's understand the requirements of equipment which will require like reagents, instruments and glassware to perform the procedure. So now let's see which type of reagent we will require in this process. So we will require concentrated H2SO4, concentrated HNO3. This concentrated acids will require for digestion process, and then phenolphthalein indicator, 1 normal NaOH, ammonium molybdate reagent, and stannous chloride. These reagents will be required for color development. The preparation of all these reagents are given in annexure. You can refer to that and you can know the exact procedure of preparing these reagents. Now let's see which type of instruments and glassware we will require to perform this procedure. So firstly we will require a spectrophotometer for taking the absorbance and readings, the digester to digest the sample, conical flask, volumetric flask, digestion tube for digestion, and the pipette for adding reagents and samples. So this was the instruments and glassware we will require to perform this procedure. So now let's see the procedure of analysis for this process. So now let's see what kind of interferences will be there in this process. So first is silica and arsenic. The positive interference can occur only if the sample is heated, and arsenate, fluoride, thorium, bismuth, sulfide, thiosulfate, thiocyanide or excess molybdate, this forms the negative interference wherever the sulfide may be removed by the oxidation with bromine water. Third is ferrous iron. This causes a blue color if concentration is more than 100 milligram per liter. Only if the concentration is this much high, the ferrous iron will interfere, otherwise it won't. For the chloride, if it presents in more than 75 milligram per liter of concentration, then only it will interfere, otherwise it will not interfere. So this was the interferences in this process. Now let's move to the step-by-step procedure. So let's start with the step-by-step procedure of measuring total phosphate into the given sample. So first we will start with the digestion process. For that we are having here two samples. First is of STP inlet and second is of STP outlet. So we will take 20 ml of each sample into the digestion tubes. Make sure you use washed glasswares for taking reagents and samples to avoid any contaminations. So here we are taking 20 ml of STP inlet and another 20 ml of STP outlet. Now we will add sulfuric acid, concentrated sulfuric acid and concentrated nitric acid into the sample. Make sure you wear gloves while dealing with the acids. So we will add 1 ml of concentrated sulfuric acid into each tubes. After that we will add 5 ml of concentrated nitric acid into each tubes. Now add few glass beads to avoid any excessive boiling while digestion. Now we will transfer this digestion tube on digester. So while digesting the sample, all type of phosphate like pyrophosphate, polyphosphate and other phosphates will get converted into orthophosphate only. So after that we will measure the orthophosphate by developing the color. That we will see in next slides. Now let's put this sample on the digester and digest till only 1 ml of sample is remaining into the digestion tube. So let's digest the samples. So after the digestion is completed, when only 1 ml is remaining into the digestion tubes, take the digestion tube out into the stand and let it cool down to the room temperature. After the solution is cooled down at room temperature, add 20 ml of distilled water into each tube. 20 ml in inlet and another 20 ml in outlet. So now we will move further in the process. I want to make here a point. In case you are measuring only orthophosphate present into the water sample, start the procedure from step number 6. Take the required amount of sample into the digestion tube and start the analysis from next step. So in next step, we will add 1 drop of phenolphthalein indicator into each digestion tubes. So we will add 1 drop in this and another in this. Now in next step, we will add 1 normal NaOH into digestion tubes till pink color is visible in it. So add till pink color is visible into the sample. So here you can see the pink color is visible. This is for inlet and this is for outlet. Now in next step, we will transfer the both solution into volumetric flask and make up it till 100 ml. So we will take this funnel and will add this solution. Wash the digestion tube with distilled water. Use another funnel for another sample. Now make up both till 100 ml mark. This is for inlet. This is for outlet. Now transfer this both solution into the conical flasks. In next step, add 4 ml of molybdate reagent into each sample. With every drop of ammonium molybdate added into the sample, this pink color will disappear. Let's see that. So you can see the color is disappeared. Add 4 ml. After adding the ammonium molybdate reagent, add the stannous chloride. 10 drops of stannous chloride into the sample. With every drop of stannous chloride added into the sample, blue color starts appearing into the water sample, which indicates the presence of phosphate into the sample. So now we will add stannous chloride. 10 drops of stannous chloride in each sample. Every drop of stannous chloride added into the sample, you will be able to see the blue color is started appear, which indicates the presence of phosphate into the sample. After the stannous chloride added into the sample, we will let the color develop for 10 to 12 minutes. Take the absorbance between 10 to 12 minutes. So now let's prepare the spectrophotometer for taking the absorbance. So after 10 minutes, when the color developed properly, you can see this for inlet and this is for outlet. The colors are developed already. Now we will take absorbance of these samples into double wave spectrophotometer. So to operate the spectrophotometer, firstly start the spectrophotometer and then calibrate the spectrophotometer. After the calibration, select the wavelength. Here we are measuring the phosphate into the sample, so we will select the wavelength of 690 nanometer. After selecting the wavelength, choose the standard curve. You have to prepare the standard curve prior. The procedure of preparing standard curve is given in annex here. You can refer to that and you can prepare the standard curve. So we will select the standard curve. After selection of standard curve, do the cell matching. So for cell matching, you have to fill distilled water into both the cuvettes for reference and for sample. Make sure the transparent side of the cuvette is clear. If this transparent side is having any type of droplets or dust on it, the absorbance reading will vary and will give errors. So make sure the transparent side of the cuvette is cleaned. So do the cell matching first. After the cell matching is done, we will measure the absorbance one by one for inlet and for outlet. So in reference solution, we will fill the distilled water as it is, and in sample cuvette, we will add the sample we have prepared already. So again, I am saying this, make sure the transparent side of the cuvette should be clear to avoid any variation into the results. So firstly, take the reading for inlet, and then for outlet. After taking the reading, we will move to the calculation part. And now let's finally understand the calculation of phosphate and total phosphorus. So basically, the formula of calculation is for phosphate is C into D, where C stands for the concentration of ortho-phosphate derived by the spectrophotometric method, and D stands for the dilution factor. So if you remember, we have taken 20 ml sample for digestion. So ideal sample taken in this process is 100 ml. So the dilution factor will be 5. That's why 2.70 is the concentration which we have derived with the help of spectrophotometer, and 5 is the dilution factor. So the answer will be 13.5 milligram per liter. This is for inlet sample. And if we want to calculate the total phosphorus in inlet, so we have to multiply this 13.5 with 0.3261. So what is 0.3261? It is the conversion factor which converts the value from phosphate to total phosphorus. So simply apply the value 13.5 into 0.3261. So that the total phosphorus content available in inlet sample will be 4.403 milligram per liter. So I have provided the value of outlet concentration to you. The dilution factor will be same 5. Calculate the phosphate and total phosphorus content in outlet and comment down so we can know if you understood the calculation or not. So now let's understand about the measurement uncertainty for ortho-phosphate. So here you can see the table. Here the given concentration of ortho-phosphate in microgram per liter, the number of laboratories which have tested the sample, and the standard deviation derived in percentage. So for 100 microgram per liter, which is 0.1 milligram per liter concentration, was tested by 45 different laboratories, and the standard deviation derived was 25.5%. For 600 microgram per liter, which is 0.6 milligram per liter concentration, that was tested by 44 different laboratories, the standard deviation derived was 14.2%. For 7000 microgram per liter, which is 7 milligram per liter concentration, was analyzed by 45 different laboratories, and the standard deviation derived was 7.6%. In case of SWA lab, the concentration of 9900 microgram per liter, which is 9.9 milligram per liter concentration, was tested, and the standard deviation, or you can say the measurement uncertainty derived is 17.57%. The source of this table is AFA 23rd edition, method 4500P and table number I. So one should always estimate the measurement uncertainty by repeatability of analysis on the standard sample of known value, so they can estimate the measurement uncertainty or standard deviation of their own facility. So this was it about measurement uncertainty. So friends, this was it about the measurement of phosphate and phosphorus in given water sample. I hope you have understand it well. We have covered everything starting from the definition to principle of measurement, equipments, procedures, and calculation measurement uncertainty. Still, you have any doubt, you can ask us in comment box, or you can follow us in Instagram. You can DM us there. We will try to answer your questions. So if you have any suggestions or any topics of videos, you can suggest us in comments, and we will try to improve our quality, and we will try to be consistent. We will be releasing the video of another parameter very soon. Till then, take care of yourself, take care of environment around you. See you in the next video very soon. Bye bye.